Hello friends, welcome again to my YouTube channel. This is exam 2, video number 1. Today we'll be talking about skeletal muscle and how it contracts, how it works. We'll talk a lot about the roles of calcium and the different types of channels and receptors and pathways and the cycle that we see inside of our muscle that causes our contraction. We will then talk about different types of muscle fibers, including fast twitch and slow twitch, and the nuances between those two. Finally, we'll be discussing how smooth muscle works and what are some of the key differences between smooth muscle and skeletal muscle. Alright, well, let's jump right into it. So first off, skeletal muscle. We're all familiar with how it functions, hopefully, because we all have muscle. Some of us more than others, but everyone has muscle. So here we have a classic bicep being flexed in my poor drawing, but just bear with me. Obviously, when we flex a muscle, what we're doing essentially is we're shortening the muscle. Now, skeletal muscle is made up of a lot of tiny muscle fibers. Muscle fiber is another word for like a muscle cell, and they have about the same thickness as hair. So human hair is actually kind of similar to what your muscle fibers are, and they're wound tightly together in groups, but they're really fine, like strings, if you will, of muscle fiber. And each muscle fiber is a group of muscle fibers innervated by a neuron, is called a motor unit. So we have what's called a motor neuron. So I'm going to draw a really crude symbol of motor neuron. That's where the synapse would be. And here's the muscle, made up of multiple fibers. So multiple muscle fibers innervated by a single motor neuron. This whole thing is referred to as a motor unit. Now let's get into how exactly our muscles work. So I'm going to do zoom up here. We have our motor neuron, and we have what the muscle here. We have some receptors and channels, and then we are going to have this big tunnel that dives deep into our muscle fiber. So skeletal muscle is often referred to as voluntary muscle because we think about moving our muscle. We can cause contractions to happen, like flexing in, pulling your fingers into a fist, stretching them out. We can do all these different things because we have control over this part called the neuromuscular junction. The neuromuscular junction is the space between our motor neuron and our actual muscle fiber. So what happens when we're sending a signal down from our brain into some sort of muscle, pick your favorite one, what we'll be doing is we'll be sending down an action potential. We've talked about that before. We'll have voltage-gated sodium channels sending down a cascade of positive charge, or the Buang, as we've referred to it before. Now, when it gets down to the end here, near the neuromuscular junction, there's going to be a voltage-gated calcium channel. And what that does is, because it's voltage-gated, when it gets the positive Buang, if you will, from the sodium, it will open, allowing calcium to come in. And what calcium does is it interact with what is known as snare proteins. There's plenty of videos on what snare proteins are, so if you're curious about that, you can go ahead and check out in a different video. But these snare proteins will mediate exocytosis, meaning these little bubbles filled with neurotransmitter. In this case, it's going to be acetylcholine, which is often referred to as ACH, will merge with the end of the cytoplasm here and will release that neurotransmitter, acetylcholine, into the neuromuscular junction. When that happens, we get acetylcholine binding to this receptor here. This receptor is known as an N1 receptor. N stands for nicotinic, and 1 is just the type, because there's multiple types of nicotinic receptors. Now the N1 receptor is connected to this channel here, which is a ligand-gated sodium channel. So when we get our acetylcholine attaching to our nicotinic or our N1 receptor, that opens up this gate and it allows sodium inside the muscle, and we get a depolarization occurring. Now this happens and it belongs, or the positive electrical field affects a voltage gated sodium channel over here, which affects a voltage gated sodium channel over here, allowing a cascade of further signal. So just to reiterate here, we have an action potential from our motor neuron coming down here, opening up our voltage gated calcium channel, which then interacts with the snare proteins, which then initiates the exocytosis, or the leaving of the bubbles or vesicles filled with acetylcholine, which is our neurotransmitter, which then attaches to our N1 receptors, which opens up another sodium channel. This one is ligand gated, with acetylcholine being the ligand, and then that does another cascade coming down here. Now when we get to this part, this tunnel is called a T-tubule, and there's thousands, millions, in fact, of these in a single muscle. Each fiber, if you're looking it up really close, if you blow it up so we're on the same scale as the muscle fiber, you'll see that it looks like somebody took a fork, a dinner fork, and just stabbed a bunch of holes all throughout the muscle fiber. And the reason for this is, when we want to contract our muscles, we want our entire muscle to contract. We don't want to wait for this cascade to slowly diffuse throughout the entire muscle fiber. We want it to happen really fast, so that way when we think about a movement, we can make it happen very quickly. So this T-tubule goes down deep into the muscle fiber itself, and surrounding the T-tubule, the little tunnel, is a membrane known as the sarcoplasmic reticulum, or we call it SR for short. The sarcoplasmic reticulum is a very special place for our muscles because it contains all of the intracellular calcium ions. So inside of this blue squiggly line, we have a bunch of calcium. And outside, throughout the rest of the muscle fiber, there is no calcium when a muscle is in relaxed state. So we have our SR here. Now, this is where it can get a little confusing, so try and stick with me. We have what is known as a DHP receptor. So a DHP receptor works very similar to how our voltage-gated sodium channels work. It basically is a voltage-gated channel, but it is not the main way to get into this sarcoplasmic reticulum. It does, however, act as a sensor to change in voltage. And why is that important? Well, we can see here that we've been causing a change in voltage cascade all the way down into our T-tubule, starting from our motor neuron down to our N1 receptor and all the way over here. So once we get to this spot, this DHP receptor will sense the positive voltage caused by the increased sodium ions, and it will pull open a second channel. Now, the second channel is not on the outside. So technically here inside of our T-tubule, that is still outside of the cell, right? It's like a tunnel from out here into here. So you can see that it's just a continuation of that extra cellular space. And so our second channel that's connected to the DHP channel right here is known as our RYR channel. RYR. And that stands for something that makes sense to a couple of people, but not very many. For our purposes, RYR works. Just know that RYR is a channel that connects the sarcoplasmic reticulum, the thing that is full of our calcium, to the rest of the sarcoplasm, which is to say the rest of the muscle fiber. So the DHP senses the positive volt, or the positive electrical pulse, basically, from our voltage gated sodium ions, and it'll pull RYR open. When this happens, because we have all the calcium inside of the cells inside of the SR, it will rush out down its concentration gradient, and we'll get a huge increase of calcium into the rest of our cell. Now, cells often, just the space inside of a cell is often referred to as a cytoplasm, but because we're dealing with muscle fibers or muscle cells, which is synonymous, we call it sarcoplasm. Sarco meaning muscle, plasm meaning space, right? So we have a bunch of calcium because it's going down its steep concentration gradient, because we have 100% calcium inside of the sarcoplasmic reticulum and 0% inside of the sarcoplasm itself prior to contraction. When we do this, we get a bunch of calcium into the sarcoplasm because we're opening up those RYR channels. And that's a mechanical gated channel. And it's mechanical because it works like a door, and the door is pulled open by our other channel, the DHP receptor, which is a voltage receptor, or voltage channel technically, but nothing's going through the DHP channel itself. It's just there to sense the change in voltage that is sent originally from the motor neuron and cascades down through our system that we talked about. And then we get what we have here. Now, calcium is the key to muscle contraction, but let's find out why. So I'm going to erase this. And in order to figure out exactly how calcium initiates contraction and is essential for it in our muscles, in our skeletal muscles, we need to kind of go over a little bit of muscle anatomy. So we have right here, let's say, a skeletal muscle. Let's continue with the bicep, since that is a pretty popular one to flex. We all know where the bicep is, right here, right? If I worked out more, you'd be able to see it better, but that's all right. So we have layers of these fibers inside of our muscles, and each one contracts. So if this is like the entire muscle here, this is a subunit, and this is a subunit, and it goes all the way down to the smallest unit of muscle that can be contracted, and that is something we call a sarcomere. Sarco, remember, that means muscle. I'm not sure what mere means, but I would be surprised if it meant like muscle unit or something like that. Anyways, so sarcomere, we have these tiny filaments, and they look like this. It almost looks like staggered ladders. And this pattern, this organization inside of our skeletal muscle, is what gives our skeletal muscle a distinct kind of striped look, because it's very organized, which is good because that makes our contraction or our movement more efficient. So I'm going to finish drawing this real quick, and then we'll go over what each part is and why it's important. Now this is just a single sarcomere. If I were to try and draw an entire muscle worth of sarcomeres, it would be like copy and paste this over here, and then over here, and over here, and it just continues down. But this is the smallest muscular unit that we see contracts. So we have a couple of things here that we're looking at. First of all, both sides where we see the zigzags, we call those Z discs. Z because it zigzags, it goes this way, goes that way, this way, that way. And then disc because, I don't know, anatomists, they like to name things. Then down the center, we have what's known as like an M line. So we have Z disc, M line, and then we have these weird looking filaments here. They almost look like little beetles. What we're looking at here, this is called myosin. And these are all proteins. So if you've ever known someone who loves to lift weights and loves to get, swole, they love eating a lot of protein. Well, muscles are largely just protein. This is a myosin protein or filament. This is the stringy thingy right here. I drew is an actin. And on top of actin, we have an overlapping stringy thingy in order to make this more accurate. So it's like a double stringy thingy. And the other stringy thingy is called tropomyosin. And there's little things on the tropomyosin, and those are called troponin. Now stay with me here. I know it gets a little complicated, but I promise once you get it down, it's easier than it looks. Alright, let's go over it. Z disc, myosin, tropomyosin is one of the stringy thingies. Actin, another stringy thingy. The little balls on here, troponin. Okay? Now these sticks and balls are just called the light chain and heavy chain. Heavy chain being the ball part, light chain being the stick part. And those are part of the myosin. And you see how they like slightly overlap here, but there's still some space here in the middle? That is the space that will shorten when we're contracting that muscle. And so if you imagine if we have, 100 of these in a row, that space starts to add up. And so if all of that space shortens at the same time, we get a bigger contraction, a stronger, more powerful contraction, and we're able to make more things happen with it. We're able to lift bigger weights or jump higher or sprint faster or so on. So now let's go back to this whole point of what we're trying to make. Remember when we were talking about the neuromuscular junction, we have the motor neuron that releases acetylcholine. That acetylcholine is connecting to the N1 receptor, the nicotinic receptor, which is connected to that ligand-gated sodium channel, opens. We get sodium creating a positively charged Wang that opens voltage-gated sodium channels that go down into the T-tubule that affect the DHP receptor that pulls the RYR channel open that allows calcium into the sarcoplasm, which causes contraction. Now, we know what our smallest unit of contraction looks like. This is what it looks like. How does calcium make this thing contract? How does it shorten this whole thing? Well, let me show you. So remember these things down here. We have the actin, and then we have the tropomyosin, and then the little balls here are known as troponin. Let's zoom in even further and look at this, because this is where calcium becomes very important. When we have actin filament, if I had another color I would use it. Maybe I should invest in that, but you know. Tropomyosin, overlapping it. This is very important because those heavy chains and light chains, the ball and sticks on the myosin, remember? Those are the things, think of them as like kind of like hands. They want to grab on to the actin, and then when we have contraction, they'll pull them over, shortening that space that was in the middle of our sarcomere, which inevitably creates the full contraction of the muscle. But it can't do that when the tropomyosin is overlapping the actin. So the tropomyosin is kind of like getting in the way of our heavy chain myosin, the ball, and then the stick part is what moves it later on, but we'll get to that. Now, the tropomyosin is on top of the actin, making it impossible for this to happen. And the whole reason why it stays there is because of the balls that were on the tropomyosin, known as troponin. So remember, troponin, tropomyosin. Troponin, instead of just being a single ball, scientists have actually found it to be 3 parts. So they just sit on top of the tropomyosin. One part, known as troponin I, it actually sticks to the actin. And then we have another part called troponin C, and that is where the calcium comes in. And then the third part which is subunit T, that is connecting to the tropomyosin. So a good analogy for this I've heard before is think of troponin like a bike lock. You're going to take your tropomyosin, which is like the chain, put it around that actin, making it so it's not able to be grabbed by the myosin itself, and then you take the troponin and lock it all together so it can't be moved. The subunit troponin I is attached to the actin. Subunit troponin T is attached to the tropomyosin, which sticks the two together, keeping them from keeping contraction from happening. And then tropomyosin subunit C is basically like the keyhole, and the calcium is the key. So troponin subunit C has 4 binding points, 4 binding spots for calcium. So when calcium comes in and is flooding the sarcoplasm, it will come in here and it will bind to what is known as low affinity calcium sites, meaning they bind, but they are only binding there because there's an overwhelming concentration of calcium in the system once we release it from the SR. There's always calcium attached to the high affinity binding sites for calcium, which are, I drew up here. Those are those ones. So there's always going to be 2 calcium ions attached to those parts. It's only when there's an overwhelming amount of calcium within the system, because our SR is flooding it, that we get the low affinity spots binded to. And when that happens, then the troponin lets go of the actin as well as the tropomyosin. And that frees up the actin to be able to be attached to our myosin head or a myosin heavy chain. Hence, contraction. And inevitably, that is why calcium is essential to muscle contraction. If you don't have any calcium released into the sarcoplasm, then that troponin is going to hang on, and that tropomyosin is going to keep overlapping that actin, and it's never going to get freed up so that myosin can grab onto it and pull it over. So that's how contraction works in a nutshell. Now, let's move on to the next part, which is what happens after the troponin falls off and we have the actin available for the myosin head. Well, let's go through it, okay? I'm going to draw it in kind of a cycle because all things physiology kind of happen in a cycle. So, myosin heavy chain, myosin head, myosin light chain, actin. This is assuming that calcium has already flooded the system and it is free to bind. When this happens, we have ATP attached. So right now, I didn't draw it very well, but right now our myosin head is still not attached to our actin filament. So what needs to happen in order to make that attachment occur, we need some energy. Well, ATP is a great source of energy. So what happens is we're going to take one of the phosphates from our ATP and we're going to turn it into organic phosphate. And the energy released when this happens is what is used to connect our heavy chain myosin to our actin. And we get ADP and an inorganic phosphate attached to our light chain myosin. And now our heavy chain myosin, or our myosin head, is attached to the actin. And it's cocked into place. So it does 2 things in this one step. 3 things if you want to think about it in terms of ATP, donating a phosphate, releases energy, cocks it into position, and sticks. Cocks, sticks. Then we're ready for what is known as the power stroke, the pulling the actin, the actual shortening of that middle part of the sarcomere, which inevitably causes contraction, right? How do we get that power from shortening the muscle? We get it by releasing that inorganic phosphate. So we have inorganic phosphate leaves the system, that breaking that bond releases energy, and we get a power stroke. And we're left with just ADP attached to our light chain myosin. But it's, you see that? It was over here, and it pulled it. Now the next step is to release, so we can do it all over again. Pull, cock, stick, pull, release. Cock, stick, pull, release. And it goes over and over again. Does this a bunch of times until you get full contraction. Now what happens in order for us to release the heavy chain myosin from the actin, we need to turn the ATP back into turn the ADP, back into ATP by adding another phosphate onto it. And once we do that, that will cause the myosin to let go of the actin, and then we're able to do it all over again. And that is how skeletal muscle contraction works from beginning to end. So that was part 1 of the video. I know you guys are like, there's a part 2. That was so long. It was so long. Now let's get into different muscle fiber types. Okay. We have essentially 2 major groups of muscle fiber. We have slow twitch and we have fast twitch. You might have heard these terms before. Slow twitch, fast twitch. Slow twitch, generally what characterizes a slow twitch muscle is it has a lot of its energy or a lot of its metabolism is focused on something called oxidative phosphorylation. Oxidative phosphorylation which means it makes a lot of ATP per glucose. So we have ATP out the wazoo for slow twitch, which is good because slow twitch is also known for its high endurance. It does not fatigue. So high endurance. It doesn't fatigue very well, I should say. It does eventually fatigue, but it takes quite a while. High endurance. Along with that, it has high blood supply. So a lot of vascularity. Oxidative phosphorylation, high endurance, high blood supply. Low power, I suppose, is the next one. So if you're one of those guys who loves to just like bench press 250 pounds, this is not the muscle fiber that you're looking for. So low power. Mostly does oxidative phosphorylation for metabolism. High endurance, so it's more of like a marathon runner's friend. And high blood supply, so we're getting lots of nutrients, lots of oxygen. That's another thing that characterizes these types of muscles fibers is uses lots of oxygen. And that's it. That's what slow twitch muscle fibers are. So another way to say slow twitch muscle is saying type 1 muscle fiber. So type 1 muscle fiber will have all these characteristics. Type 2 muscle fibers, there's 2 subunits of type 2. There's type 2A and what is known as type 2X. But a lot of people are starting to call it type 2B because we're starting to find that it's a completely separate thing from type 2A. But let's just go over the general characteristics of what a fast twitch muscle is. And you could probably guess it's pretty much the opposite of everything that we put for slow twitch. So fast twitch mostly uses glycolysis as its metabolism. So it is very low efficiency when it comes to ATP per glucose molecule. It's a net plus of only 2 ATP, whereas oxidative phosphorylation is way more. It's up there. I don't have it memorized, but it's a high number, like way more than 2. So fast twitch, low efficiency with glycolysis. which means it has low endurance. It fatigues very quickly. It has low blood supply, so not a ton of vascularity. In fact, it would be what we would refer to as white meat. So if you're comparing like a nice bit like smoked brisket, that is going to be slow twitch muscle from the cow versus a very like tough chicken breast that's just like white. There's no juiciness to it whatsoever. That is fast twitch muscle, and so it'll appear whiter because there's less blood supply. High power, very high power, and for athletes that's good, right? And does not use oxygen. The type 2X doesn't need oxygen really that much because it uses just glycolysis. Type 2A has some oxidative phosphorylation. It's kind of like an in-between the two. It's not super slow twitch and it's not super fast twitch. Type 2A has a little bit of the characteristics that we see with slow twitch, but it's still more fast twitch than it is slow twitch. So it'll be more towards this side. But for type 2X, it's definitely 100% fast twitch. So we got low oxygen here. And that's kind of like the difference between the two. Remember, type 2A is what I like to know, like think of it kind of like the Goldilocks muscle fiber or fiber type, because it's not too slow, not too fast, it's right in the middle. So with fast twitch, with glycolysis, you get like 30 seconds of power before your muscle fatigues. If you've ever been in the gym where you're trying to lift pretty heavy weights, say you're doing a bicep curl with a significant amount of weight, you can only get to so many before you feel your muscles just relax and release. And that's because you reached, well, that's actually because of something that we're going to talk about next. But anyways, those are the two main types of muscle. We talked about the nuances between type 2A and type 2B, as well as what they do. And everyone has a mix of both. Nobody has 100% slow twitch. Nobody has 100% fast twitch. You need a mix of both to be able to function like a normal person. Okay, moving on. Let's talk about reflexes real quick. I'll try and make this quick because this is already a really long video. I apologize to anyone who has to watch it. So muscle reflexes. Reflexes are something that we don't have to think about. They're kind of involuntary, which is really nice because when you're in say a game of soccer or basketball, sometimes you need these to happen so that you don't injure yourself. It's kind of your body's safety net to keep yourself from, tearing tendons or breaking bone even because of the strength of our own muscles. So this is our body's way to keep us safe. So let's say we're lifting that really heavy weight doing bicep curls. Let's say it's like, 50 pounds. We're trying to pull it really hard, right? We're at the gym. That's a massive bicep, but and a really small forearm. But we're pulling it up. We're pulling, and let's say we're just at that point where we're not really moving our arm anymore, but we're just keeping it there while we're still trying to contract our muscle. This is where something really interesting happens. Inside of our muscles, particularly towards the end in the tendons, the tendon is the part that connects muscle to bone. So usually when we contract our muscle, it's shortening the muscle body, right? And that pulls the tendon, and the tendon, since it's attached to the bone, will then pull the bone. So muscle shorten, pull tendon, tendon pulls on bone, and we get movement. However, if we're dealing with a significant amount of resistance so that our bone is no longer moving, but we're still shortening that muscle because we're sending down those action potentials into our motor neurons, then the only thing that can move, or the only thing that could have strain on it, is our tendon, the thing that connects the muscle to the bone. And when this happens, it stimulates something known as your Golgi apparatus tendon. So Golgi apparatus tendon is kind of like a sensor that our body has inside of our tendons that will be able to sense that strain, that pull that we're putting on the tendon. Now, our muscles are very, very strong. And if we didn't have that Golgi apparatus tendon, then we could contract so hard that we could either tear our muscle from the bone or we would break our bone. But in order to keep us from doing that, because that's horrifying, our body sends a message through an afferent pathway to our spinal cord. It doesn't even go up to your brain because you don't even think about it when this happens. It's just a reflex. From the Golgi apparatus, it sends an afferent message to the spinal cord, which synapses with an efferent neuron coming from the spinal cord. So it just directly goes to the spinal cord and back, and it tells the muscle to stop. Because essentially it's like, whoa, we're putting a lot of strain on this tendon. Something bad is going to happen if we don't stop this movement, because we can either tear something or break something. So it sends it over here, and the reflex is, oh, okay, we need to relax this muscle. We need to keep it from tearing or breaking something. And that's why, if you've ever had that experience where you're just like really trying hard and you can't even move your arm or your leg or whatever you're using to lift a weight, you'll have a moment where you feel like, oh, I have no strength left. There's nothing left in the tank. That's actually not true. You have plenty left in the tank. But because of this reflex, your body kind of forces you to relax that muscle. And at the same time, it's sending another neuron to the muscle that does the opposite movement, so in this case the triceps, and it tells it to contract. And why that happens, you're pulling, and it's like, relax this muscle, the bicep, the one that's putting a lot of strain on that tendon, and contract the triceps. So what's going to happen? You're going to get the opposite of what you've been trying to do. So that's known as the Golgi apparatus tendon reflex. It keeps us safe because we're stronger than we know. It sounds kind of like a backhanded motivational speech, but that's how it is. There's two other reflexes that I'll talk about before we get to smooth muscle. We have Golgi apparatus, and then we have these twos. What is commonly known as the bag and chain. I'm going to go really fast here because this is taking a long time. So we have the chain and the bag. The chain is, both of these are what are known as intrafusal muscle fibers, which means they're purely for sensory purposes or reflex purposes. But what they do is the chain will sense a stretch. They'll both sense muscle stretching. So if you've ever had the excruciating experience of doing pull-ups, I'm sorry. But once you're up here, you can either let yourself go down really fast, just like a big drop, but an even harder thing to do is to let your body go down from the pull-up very slowly. When we do that, you're still working that muscle, the latissimus dorsi, as you're letting yourself go down, but it's also stretching the muscle. And so you get what's known as an eccentric contraction, stretching while you're still kind of trying to hold it. You're trying to control the movement. So we have these two intrafusal muscle fibers that sense that stretch when it's happening. And what's going to happen when we're stretching a muscle is we're going to have the chain fiber, the like chain marks, the black spots, those are just all nuclei. Bag fibers, obviously they have like the bag-like formation in the center, but that's where all the nuclei are like kind of compiled together. So chain fibers, they'll have sensory for duration of a stretch. So we'll be able to tell, oh, this is a really long stretch. Maybe we should do something about it because it doesn't seem like anything's changing anytime soon if you're stretching for a really long time. The bag fiber, recognizes the frequency of stretch. So it'll be like, hey, we're getting quite a bit of stretch. It's happening a lot. And yeah, we need to do something about it. So what happens when these two muscles that are found in the body of your muscle, so in the center of your muscle, you have these sensory fibers. What they'll do is they'll send through an afferent pathway, once again, similar to the Golgi apparatus, to the spinal cord, like that. And we'll get a reflexive response to that muscle to tell that muscle, hey, we're stretching a lot. Let's try and not stretch. Because if you stretch too much, then that can be bad. You can get a muscle tear. And remember, this one's in the body of a muscle. So Golgi apparatus was in the tendon. So if we have a muscle, Golgi apparatus would be in the tendon part. The bag and chain would be in the muscle fibers within the body. So if you're stretching the muscle body, then these will be stimulated. You'll get a reflex that causes that stretch to actually turn into a contraction, and it will become stiffer, and you won't want to stretch as much. And this is why stretching, especially before you're warm, is painful, because your body's kind of like sending reflexes that are fighting the stretch. Alright, well done. Last thing we need to talk about, smooth muscle. Smooth muscle. Everything before the neuromuscular junction that we talked about with skeletal muscle works the same. Action potential down to the motor neuron. Motor neuron releases a neurotransmitter, and that's where we get the difference, right? After that, because instead of our N1 nicotinic receptor on smooth muscle, we have a G protein. So we're still releasing through our vesicles that are being fused with the snare proteins that are activated by the calcium, which is coming in through the voltage gate. Calcium. Because of an action potential. That's all the same. But instead of an N1 receptor here, we just have a G protein. Acetylcholine acts as ligand, activates G protein. G protein activates a second transmitter known as IP3. IP3 goes to the sarcoplasmic reticulum, because smooth muscle have those as well. Opens up channel to release calcium. See how that works? Now notice there is no T-tubule in smooth muscle. This is because unlike the skeletal muscle, which was very structured, where we had the Z lines and like the kind of staggered look with the myosin and actin and the different filaments that were making up the skeletal muscle, smooth muscle is not organized like that. And that's what gives it the smooth appearance, because it's all kind of like jumbled up, webbed up in there. So it doesn't really matter that if we release all the calcium at the same time. In fact, we want different sections of our smooth muscle to be contracting at different times. Think of your intestines. If you have your intestinal tract, you don't want the entire intestine contracting at the same time. That'd be really bad, and that would make digestion basically impossible and extremely painful. Instead, we have contraction that kind of just goes throughout. There's different parts that are what are known as in phasic phase, and there's other parts that are in latch stage. Latch stage is basically contraction. Phasic stage is basically not contraction, but it's not turned off. It's never completely relaxed. It's always working. And so, once again, everything's the same before we have the neuromuscular junction. The only difference is down here, this receptor, instead of being an N1 receptor, we have a G protein that then, when activated from a neurotransmitter, activates our IP3, which then turns on or opens up our calcium voltage gate channel from the SR. Then we have calcium rush in. Now, calcium works a little bit different in our smooth muscle because, like we said, we don't ever want to turn off smooth muscle. Smooth muscle are vital organs like our intestines, our kidneys, our liver, our stomach. We don't want to turn that stuff off. We want it to always keep running. And so, what our calcium does, we still have our heavy and light chain myosin. We still have our actin, but there's no tropomyosin and there's no troponin, because in skeletal muscle, those are the things that basically relax or turn off our skeletal muscle. Remember, we don't want to do that in smooth muscle. That would be really bad. We're always going to have a phosphate there, right? Calcium comes in and it activates this thing called calmodulin. Calmodulin kind of looks like the troponin subunit C, where it has 4 binding sites for calcium. And once calcium attaches to our calmodulin, our calmodulin then attaches to what is known as our MLCK. MLCK stands for a really big word that I'm not going to go over right now because this video is already 52.5 minutes long. MLCK looks kind of funny. It looks like a snake eating itself. This is the activation site. That's the tail. So it kind of inhibits itself most of the time until our calmodulin is activated by calcium that is released from the SR. And then that calmodulin connects to that tail. Once it connects to that tail, our MLCK is no longer inhibiting itself because its tail is already being taken up. And now it's free to move another phosphate, inorganic phosphate, onto our myosin light chain. And that creates the energy necessary to begin contraction or attachment from our myosin heavy chain onto our actin. We have a breaking of bond through hydrolysis, power stroke, and it does a very similar cycle as the skeletal muscle. But the big difference between skeletal muscle and smooth muscle is, first of all, there's no T-tubules in smooth muscle, and there's no N1 receptor. It's going to be a G protein, which connects to IP3, which opens up the SR, which makes calcium go into the sarcoplasm. There's no tropomyosin. There's no myosin. Or there is myosin, sorry. There's no tropomyosin. There's no troponin, because there's no need to turn off smooth muscle. We want it to constantly go. Latch phase is when it's continuing to stay in the contracted conformation. So when it's after the power stroke, it's still connected to that actin. It's going to hold until ATP reattaches it to it, and then it'll let go, and it'll start over. And essentially, that's how smooth muscle works. Thank you for watching my video. Hope you had a great time, and I'll see you next week.